Hi and welcome to Unit 5, Bacterial Growth in BIO193A. And we are going to talk about microbial growth and particularly bacterial growth, which when we refer to growth in this context, we are talking about growth in numbers, not so much growth in size. How do bacteria divide? You may recall from previous classes that eukaryotic cells divide by mitosis. Prokaryotic cells, such as bacteria, divide by a process called binary fission. So fission is breakage, and binary basically means breakage in two, which is the really clear definition of what happens. So as you can see from the diagram below, the cell starts, replicates its DNA, and then there will be a division forming inside in the middle of the cell. That's called the septum. You see that together with this, the cell will become a little bit longer, and the two DNA molecules, remember that DNA in bacteria is circular, kind of separate from each other into each of these parts or halves, which will eventually separate and become two new cells. So let's say that we inoculate bacteria in a broth, and you want to follow how they grow. So in this scenario that we inoculate bacteria in a broth and leave them alone, you will be able to observe four phases of this growth, which are the lag phase, the log phase, the stationary phase, and the death or decline phase. So what is the lag phase? As you can see from this graph, which is plotting time versus the number of bacterial cells in logarithmic amounts, in this first phase, it's flat. So that means that there is no increase in the cell number. Lag phase is often defined as preparation for growth. So for example, if you take a bacterium from one medium and place it in a different medium, the bacteria may not have the enzymes necessary to be able to process those nutrients. So it needs some time to produce those enzymes. Think about what we learned about operon. So it needs some time for the operon to be turned on, for the enzymes to be produced. Another scenario could be endospores. So this germination process of the endospores takes some time for the bacterium basically to wake up and be ready to go. The length of the lag phase is going to depend on the scenario. So for example, if you are just placing bacteria from old medium to new medium, but it's the same kind of medium, then the lag phase is going to be very short because there is no real adaptation to be made. And it can be longer, for example, in the endospore case. After that, we have the log phase, and log comes from logarithmic. So there is this exponential increase in number of the living bacterial cells. So these are your happy bacteria, continuously dividing by the process of binary fission we saw before. However, again, we are not adding or removing or doing anything to that culture, so eventually the amount of nutrients available will decrease and the number or the amount of waste products will increase, which means that some cells will start to die. And now we are going to see a plateauing of the growth because the rate of the cell division and cell death will be roughly equal. So this is what we call the stationary phase. And eventually, if you don't do anything to that culture, the food will run out. There is just too many waste products around, so cells start dying. And this is a little bit of a mirror image to the log phase, death or decline phase. It's an exponential decrease in the number of living bacterial cells. Now you may wonder, how do we know these numbers? How can we generate these kind of graphs? Well, you can use as a proxy the cloudiness of the culture. So cloudiness is the same as turbidity. And there is an instrument called Spectral Photometer, which you see to the left. And what Spectral Photometer does is to shine a light through liquids. And you can select the wavelength of light that will be absorbed by whatever you want to read. So in this case, we are looking at bacterial cells, and light will be absorbed by bacterial cells at 600 nanometer wavelength. So if you see so let's look at the upper diagram here. You have the light source. The light goes through a prism, which is going to separate the light into different wavelengths, and then through selectors, only that 600 nanometer wavelength will be allowed to pass through. And in this case, in the upper example, this is an empty broth. So the light will just go through. There will be no light absorbed. So you're going to get this value low optical density, or also called absorbance. So remember your graph, how it started, that lag phase, it's kind of a low number. This is because you have very few cells to measure. Contrast what happens in the second example. So here you have a broth that is clearly very turbid. So a lot of the light gets absorbed or refracted by the cells in the broth. So the amount of light that goes through to the detector is smaller. So now the number that you see is much higher. So we are looking at a high OD or absorbance number. Now don't imagine a level of 200. So high absorbance would be about, let's say, 1 or 2. I don't think that spectral photometers read much higher, above 2. Okay, so in real life, what you do to measure this is to start from a single colony. You put it in a small amount of broth, let's say 10 milliliters. You culture it overnight, so then you get the culture going, and then you take a larger volume of broth, let's say 250 mils. You inoculate let's say 5 mils, so half of that original tube, into this larger volume of broth, and then you start reading optical density. So you take the optical density of 0 hours, which we expect it to be quite low, and then every 30 minutes you will take a small amount, let's say 1 mil of the broth, and measure the optical density. And you keep doing that until the reading becomes static, meaning it's going to plateau. The word aliquot means taking a small amount. So aliquoting something means you dividing whatever it is into small quantities. And then you are going to obtain a graph in time. And let's just show you, this is real data from an experiment that was done in a class. So the same bacterial culture, I believe this was E. coli, was placed in the same medium and then incubated at different temperatures. And then you were measuring the optical density at, actually here we were looking at every 10 minutes. So you can see on the right the legend of the different temperatures, and you can see how in some cases it didn't increase very much, in others increase much higher. So if you were asked which is the best temperature, clearly you have to look at the graph that goes up the highest, and that would be 55 degrees. So E. coli can grow very well. I mean, it grew well at 35 also. 35 and 45 was pretty much the same, but it didn't grow very much at 15 or 25, which is kind of coolish. We are going to look now at another very important factor in microbial growth, which is their oxygen requirement. And what this diagram is showing you is the patterns of growth of bacteria when inoculated in, let's say, an agar deep, or can be special broth. We are looking mostly at soft agar broth. So in a tube, which is full of this medium, the closest to the top is going to have the highest oxygen concentration, and the bottom is going to have the lowest. So you will be able to see these different patterns in accordance to oxygen requirements. And I'm going to start with the first and the fourth, which are the most obvious ones, the obligate one. So obligate aerobes require oxygen. Without it, they die. Obligate anaerobes die in the presence of oxygen. So you can see that obligate aerobes grow basically only at the very top of the tube where there is the most oxygen, and conversely obligate anaerobes are going to grow in the bottom. Now number 2 and 3 are very similar, but they are slightly different in their preferences. So number 2 is facultative anaerobes, and very often students get confused about these facultative anaerobes. So what it means facultative is that's the choice, or not a choice, the option. So facultative anaerobes means that they can grow both aerobically and anaerobically, but they do prefer oxygen. So they survive without oxygen, but they do prefer oxygen. So if you look at the growth pattern, you see that they are everywhere, but there's a little bit more of them on the very top. However, aerotolerant anaerobes, they don't use oxygen at all. They are indifferent to oxygen. So you see that the growth pattern is completely symmetrical compared to the facultative anaerobes. And finally, we have a special group, which is called microaerophiles, which use oxygen, but only at very low concentrations, or lower than the atmospheric oxygen. So they are going to form this ribbon away from the top, but not completely at the bottom. So this is just a special group of bacteria, and Campylobacter, which is one of the GI condition-causing bacterium, is a microaerophile, so that requires special incubators. So let's look now at these growth patterns in real life. And the broth you see on the picture is called the thioglycolate broth. So this is a broth, but it also has a very low amount of agar. So it's not like just liquid broth. It has a little bit of consistency to it. And what thioglycolate does is that captures oxygen. So that means that towards the depths of the broth, there's going to be very little oxygen. So it allows the growth of anaerobes. Okay, so let's look at the examples. A is E. coli. So these are facultative anaerobes, so you can see that there is growth through the tube, and although it's kind of hard to see, but there's a little bit more on the top. B is Clostridium. So this is an obligate anaerobe, and you see that it only grows away from the top. Number 3 is Staph aureus, which is also a facultative anaerobe, and you can see the growth through the tube and a little bit more also at the top. Now, if you wonder why it looks so different from, let's say, number 1, this has to do with motility. So when motile cultures tend to be kind of all spread out in the agar in the broth, and the Staph aureus, as you can see, is just growing very located to it where it was inoculated. So it probably was inoculated with a needle or with a loop, but most of the growth is very limited to that place. Neisseria sicca, this is a microaerophile. You can see very clearly that there is this ribbon of cells kind of, let's say, in the middle. So it's not aerobic, it's not anaerobic, but it's very localized to a certain value of oxygen. And then we have obligate aerobes, which would be for example, Pseudomonas. So you see it can only grow, or mostly grows in the top. You can see a little bit of cells in the bottom, well, not much here, but when they become older, they start dying and then they may sink, but the growth will be seen at the very top. And changing completely the conversation here, I'm going to talk about viruses. So you recall that viruses are not alive, so they need to infect other cells and hijack their machinery in order to replicate. So you cannot put viruses in a broth and grow them, but you can put viruses in a broth with cells, and then they would infect the cells. So how you detect that there are bacteria? So there is an assay that it's called the Plaque assay, and this is particularly useful when you are looking at phages or bacteriophages or phages. It's sent both ways. So these are viruses that infect bacteria. So what you are seeing on that plate is that you have a bacterial culture. So this is what we call a bacterial lawn. All the bottom of the agar is covered with bacteria, but where viruses are present, they would infect the bacteria and kill them. And then you see this clearing. So each of these spots is it's kind of the inverted image of a colony. So this would be the response of viruses killing the bacteria. So instead of bacterial growth, you're seeing bacterial death due to viruses. So this is, so you report PFUs, which means plaque-forming units. You remember how when you were counting colonies, you were referring as CFUs, colony-forming units. So kind of the same way you will refer to PFUs per mil, based on the original sample when you are reporting the number of viral particles. And that was all for today. Thank you very much.